Okay, so now we are live on YouTube. Thank you, everybody that's once again came to watch another installment of the Immuno webinars. Today we are going to have Dr. Lucas Bolini doing the honors of introducing our special guest, Professor Sophie Laffont. Lucas, please.
Good afternoon, everyone, and welcome to another edition of our Immuno webinars. Today we have the honor and the privilege of having here with us Dr. Sophie Laffont-Pradine. Dr. Sophie got her PhD in immunology under the supervision of Professor Jean-Charles Guéry at the University of Toulouse, after which she spent a period in Oxford as a postdoc. She then came back to France, to the University of Toulouse, where she currently is a researcher at the Center of Physiopathology in Toulouse-Bourbon, at the Institut National de Santé et de la Recherche Médicale, or the National Institute of Health and Medical Research, associated with the CNRS. She has currently an impressive body of work regarding sex differences in immunity, which have yielded over 2,000 citations according to Scopus, and an H-index of 22. So thanks again to Dr. Sophie for accepting our invitation, and the floor is yours, and sorry if I butchered any French.
That was perfect. Thank you very much for the kind introductions and for the kind invitation. So I'm going to share my screen. Let me know if you can see it. Yeah, does it work?
Yes.
Is it full screen for you? Yes?
Yes.
Perfect. Thank you. So I'm just going to... Okay, so again, good afternoon, everyone, and thank you very much for the kind invitation. It's my real pleasure today to present some of the recent work we are doing in the lab. So as Lucas mentioned, in the lab we have a long interest in trying to understand the cellular and the molecular mechanism underlying sex differences in immunity. Indeed, it is now well accepted that there are sex differences in immunity, and that in general, women, they mount stronger immune responses than men. So this can be easily illustrated, for example, for infection, where women in general have a higher capability to control infection. This has been shown during the COVID-19 pandemics. Indeed, also, although men and women can be infected at similar rate, we can see that men are more likely to develop severe COVID and more likely to die from COVID, and this has been shown worldwide. It has also been demonstrated that there are sex differences in cancer. The incidence of cancer is higher in men, and the likelihood to die from cancer is also increased in men. However, the price to pay for women for this strong immunity is autoimmune diseases. Indeed, you can see here that, so I'm going just to increase. So you can see here that there are some autoimmune diseases listed. These are the most 10 common autoimmune disease listed, and you can see that for most of them, women develop the prevalence is higher in women, and for some diseases, women the disease is mostly a woman disease. This is the case, for example, for SLE, where you can see that 90% of patients are women. So, of course, many factors can explain those differences in immunity. We can consider gender, behavior, and constructs, but in the lab, we are trying to understand how biological factors can influence this immunity. And by biological factors, what we mean of course, sex hormone, so the impact of X chromosome, X-linked gene on immunity, and also the impact of sex hormone, such as estrogen and androgen on immunity. So here you can see that this is the X chromosome. You can see that the X chromosome is really enriched in immune genes, for example, TLR7, TLR8, but also FOXP3. And we can assume that this high concentration of immune genes can have implication on immunity and sex differences in immunity. So in the lab, what we are trying to understand is how both sex hormone, but also sex chromosome can, through the direct intrinsic control of different actors of both of innate and adaptive immunity, can explain and can participate to these sex differences in immunity. Okay. So I am going briefly to show you some results. So the first, briefly, because this has been published quite a long time ago, briefly how sex hormones can control immunity through the demonstration that TLR7 can escape X chromosome inactivation and can therefore have an impact on immunity. And the second part of my talk would be on the impact of sex hormone on a subtype of innate cells, which are called ILC2s, for group 2 innate lymphoid cells, and the consequences it can have on asthma. So to balance X chromosome gene expression, there is something called X chromosome inactivation. Indeed, during embryogenesis in female cells, one of the two chromosomes, either from the paternal or the maternal X, will be randomly inactivated. So in some cells, it will be the paternal X chromosome that will be expressed, or the maternal chromosome that will be expressed. And this is leading to a mosaicism within women, as 50% of the cells of women express chromosome coming from the father, or 50% of the cells express the X chromosome coming from the mother. But this is not a perfect mechanism, because we know that 25% of the X-linked gene can escape X chromosome inactivation and can therefore be expressed within 2 copies in some cells. Okay. And as I mentioned before, we know that a lot of gene related or associated to immunity are expressed on the X chromosome. And as I said before, TLR7 is expressed on the X chromosome. And we know that TLR7 play a key role in lupus development, in SLE development, because it has been shown that TLR7 overexpression in mice can lead to spontaneous lupus, but also that the deficiency of TLR7 in lupus-prone mice can protect from SLE development. So this is suggesting that indeed TLR7 is implicated in the development of lupus. So we ask the question whether TLR7 could escape X chromosome inactivation and can therefore contribute to sex differences immunity by being expressed at double copies in female cells. So this is showing women cells. So and this is in red you have TLR7 expression, and in blue you have the this which is the long non-coding RNA, which is going to go on the X chromosome that is supposed to be inactivated. So you can see that you have when you look in a women, when you look in, for example, in B cells or in plasmacytoid dendritic cells, which are expressing high level of TLR7, you can see that you have monoallelic cells, which means that you have cells that express only one TLR7 copy from the X chromosome, the active X chromosome, and from the inactive X chromosome, you don't detect any TLR7. So you have indeed one copy of TLR7, which is normal because the TLR7 should be inactivated. However, we found that some cells will express two copies of TLR7. The copy coming from the active X chromosome, but also the copy coming from the inactive X chromosome, which means that TLR7 can escape X chromosome inactivation and can therefore be expressed in double copy in female cells. So we have shown that this is true we have tested many donors, and we have shown that this is true in different types of cells where TLR7 is expressed in women, so in B cells, in monocyte, in plasmacytoid dendritic cells. So this is different female donors, and you can see that you have approximately 30% of cells that will express two copies of the TLR7. And what we have found is that, for example, in B cells, that this inactivation this escape from X chromosome inactivation is leading to an higher expression of TLR7 in those cells, both at the mRNA level, but also at the protein level. And what we have shown is that the B cells expressing two copies of TLR7, they will proliferate more, they will differentiate more towards plasma cells, they will have higher class switch. So basically, in women, you have a special type of B cells that are endowed with special capacity to produce cytokine and to produce immunoglobulin. So, and we think that the presence of these B allelic cells in women can contribute to SLE development, and we are actually developing mouse model to demonstrate this hypothesis. So now I will move to to the second part of my talk, which is dealing with asthma. Indeed, there are sex differences in asthma. And so here you have the prevalence of asthma and the hospitalization leading to asthma. And you can see that while during childhood, boys in blue are more likely to develop asthma, there is a switch around teenage age. And now after 20 years old, you can see that women, they are more likely to develop asthma, while you have a neat amelioration of asthma in boys. So this switch around puberty is strongly suggesting of a role of hormones in the disease. So we analyze whether these differences shown in asthma in patients can also be seen in asthma models. So this is one asthma model we use, but we also verify that in other type of asthma models. This is a model where mice are given azoxystrobin by inhalation, and we simply compared male and female mice, and you can see that after asthma development, you can see that the lung of female are more inflamed. There is an increased infiltration of CD45 positive cells. There is an increased number of Th2 in those female, and also an increase in IgE. So we can recapitulate the phenotype seen, for example, in patient where there is in female the severity of asthma is higher as compared to male. So we try to understand those differences and what if hormones can explain those differences. So first, I will just briefly explain the pathophysiology of asthma, which has been a bit revisited recently with the discoveries of ILC and ILC2s for group 2 innate lymphoid cells. So those ILC2s, they are part of the ILCs family. They are innate type of cells. so that they don't express TCR, BCR. They are of the lymphoid families, but they will express rapidly and a huge amount of cytokines. And the ILC2s from the group 2, they will express a lot of type 2 cytokines. Indeed, we know that when there is a parasite or an allergen, for example, in contact with epithelial cells, those epithelial cells that will be damaged, they will release alarmin, such as IL-33, and this alarmin that will activate ILC2s. Those ILC2s that are located within the lung, they will proliferate a lot and produce a lot of cytokines, such as IL-13, IL-5, and those cytokines, they will contribute to asthma development because they will activate eosinophil, they will induce mucus production, they will induce muscle contraction, and then we also induce through by promoting the recruitment of dendritic cells through the lymph node that will promote Th2 cell development. So we know now that those IC2s that are really key in the first phases of asthma. They are key in inducing asthma, but also in perpetuating asthma. And this has been shown in a mouse model of asthma that deficiency of ILC2s is associated with less asthma development. And we also know that those ILC2s seem to be important in asthma in patients, because in severe asthma patients that increase in quantity, there is a correlation of asthma development and their present. And we also know that in asthma patients, there is an increase in their capacity to produce IL-5 and IL-13. And also, GWAS studies have shown that genes related to asthma development, ILC2's development or ILC2's function, such as GATA3, ROR-alpha, IL-13 or IL-33 receptor, which is the receptor for the alarmin, is associated with asthma development. So ILC2s are really important in asthma development. So we ask the simple question first: could ILC2s be regulated by sex hormones and therefore could contribute to sex differences in asthma? So we started with a really simple experiment. First, we compared the lung of male and female black 6 mice. So this is the strategy, the gating strategy we use to analyze ILC2s in the lung. So they are hematopoietic cells. They don't express lineage marker. They express the GATA3 transcription factor. And what we showed is that indeed, when we compared male and female lungs, we can see that the female lungs are enriched in ILC2s, both in frequency and in number. And this is not only true for the lung, but in every single organ where ILC2s can be found, we saw that there was an increase in their frequency in female organs. So this was true for the fat, for example, where you have a lot of ILC2s, and also in different types of lymph nodes, such as the mediastinal, but also the mesenteric lymph node. So these differences in frequency is also coming with differences in phenotype. So most of the although most of the markers of ILC2s are not different, we could see that there was some differences in KLRG1 expression, so I will come back to that later, and also in ST2 expression. And also what we noted is that the capacity of ILC2s from female to proliferate was increased, as shown by the Ki-67 expression marker. So after that, we said, okay, so there is an increase in the ILC2s in female tissue. Could this be due to hormone? So we first tested the impact of estrogen. So what we did, we compared the frequency of ILC2s in the lung of male, female, or female that have been ovarectomized. So we took out the source of estrogen in these mice. And what we could see that basically there was no impact of ILC2's frequency. We also analyzed the frequency of ILC2s in females that do no longer express the estrogen receptor alpha within the hematopoietic compartment using the Ty2-Pre system. And in this system, again, we couldn't see any differences. So from that, we concluded that estrogen has no impact on ILC2s. And we confirmed that by showing that that females that are overectomized or not, they have the similar susceptibility to allergy development. However, now when we analyze the impact of androgen on those ILC2s by comparing the frequency of ILC2s in the lung of male, female, or male that have been castrated, now we could see that male that have been castrated, the frequency of their ILC2s are not similar to those that we find in female lungs. So it seems that androgen, they exert a negative impact on ILC2s. So we went further on that, and we decided to evaluate whether indeed ILC2s express the androgen receptor, because to have a direct impact of androgen on ILC2s, they have to express the androgen receptor. So we compared first by mRNA expression, the expression of the androgen receptor here, but also the expression of estrogen receptor alpha, beta, or the progesterone receptor in ILC2s coming from different tissues, so the bone marrow, the fat, the gut, the lung, or the skin. And this is the glucocorticoid receptor, by the way. And what we could see is that indeed in ILC2s, they do not express estrogen receptor. So this was maybe the explanation why we couldn't see any impact of estrogen on their frequencies or number. However, they express high level of the androgen receptor, similar level to those of the glucocorticoid receptor. So this was by RNA expression. So we decided to verify if the protein was expressed. And indeed, we could confirm that in a wild-type mice, the androgen receptor was expressed. So this is done in ILC2s that have been sorted from the lung of mice. And we could see that indeed ILC2s, they express the androgen receptor. This is the control, the tested extract. However, we have in the lab androgen receptor knockout mice, and now the androgen receptor was no longer expressed. So the androgen receptor is expressed. Is it functional? So to verify that the receptor was functional, we use mice that are able to report the activity of the androgen receptor. So indeed, when the androgen receptor is a nuclear receptor, so after the ligation of the hormone to the receptor will translocate to the nucleus and will associate to androgen responsive element to mediate its effect. And in this mice, the luciferase gene is under the one of the luciferase has been androgen receptor, element have been put just before the luciferase promoter. So when the androgen receptor has linked the testosterone, it will the luciferase gene will be active. So what we could show is that in these mice, we injected IL-33 to have ILC2s to proliferate and to be able to purify ILC2s. So we purified ILC2s from these mice. And when we tested the luciferase activity in ILC2s, we can see that indeed we can detect luciferase activity specifically within sorted ILC2s. So it means that not only the receptor was expressed, but also the androgen receptor was functioning. So androgen receptor is able to mediate androgen-mediated effect within ILC2s. So we then decided to evaluate to generate mice to directly prove that indeed androgen receptor within ILC2s are important to control the ILC2's frequency. So we generated mice selectively lacking the androgen receptor in the lymphoid compartment using pre-mice using IL-7 receptor alpha pre-mice. So in these mice, the androgen receptor will be deleted in mice expressing the IL-7 receptor alpha. So this is not a perfect model because, of course, ILC2s are not the only type of cell expressing the IL-7 receptor alpha. But I mean, there is no, so far, there is no really perfect ways to directly target ILC2's deletion. So this was at least at that time a not so bad model to use. So we decided in these mice to first we evaluate indeed that we first check that indeed in these mice we have a deletion of the androgen receptor. So these are the control mice, the flox mice, and these are the mice. These are the mice lacking the androgen receptor. So you can see that we can detect the androgen receptor in ILC2s from the control mice, but we have a perfect deletion of the androgen receptor in ILC2s from the IL-7 pre-mice. And in this mice, so these are the control mice. This is what I've shown you before. Basically, that in control mice, you have increased ILC2's frequency in female tissue. So this is in the lung. But now you can see that in the androgen receptor knockout mice, specific the knockout within the lymphoid compartment, you have no longer sex bias in ILC2's frequency. So we have similar results in asthma models. So now when you take mice selectively lacking the androgen receptor in the lymphoid compartment, there is no sex bias in asthma development. So it means that androgen, they will negatively impact ILC2s in males, and this is likely to explain the sex differences in asthma severity we saw before. So we decided to move forward and to try to understand how androgen were inhibited ILC2s. So I've shown you that ILC2s frequency and number are inhibited by testosterone, but we also shown that testosterone androgen are able to limit ILC2's production of cytokines. So IL-5 and IL-13 production is decreased in ILC2s by males. So we decided to understand how testosterone can have this impact. So testosterone can act on ILC2s within tissue, but also in ILC2s in the bone marrow. Indeed, ILC2s, we know that they will populate the lung during the perinatal period. And during adulthood, there is really few ILC2s coming from the bone marrow, and most of the ILC2s that will proliferate during adulthood, they are the ones that basically came during the perinatal period and that proliferate in situ during the adulthood. So we decided first to test whether testosterone could act directly on those ILC2s that are resident in the lung. So to do so, we decided to we took female mice, and those mice, they were treated with testosterone for 10 days. So they are females, there are really few testosterone circulating. So we give them testosterone and we analyze in the lung the frequency of ILC2s. So these are the results at steady state. So non-manipulated mice, mice only given testosterone. So these are male, these are female mice. females that have been overectomized or females that have been overectomized and given testosterone. And you can see that only 10 days of treatment with testosterone can reduce the number of ILC2s within the lung. And now we have similar number of ILC2s in the lung of male mice or female mice treated with testosterone. So this is a steady state. Is it also the case during inflammation? So mice have been treated with IL-33. So the alarmin that is released when the epithelial cells are damaged. So we can mimic asthma development by directly injecting IL-33. And you can see now that this is also the case during inflammation. During inflammation, you can decrease the frequency of ILC2s in female mice by giving them testosterone. This is associated with decreased ILC2, but also with decreased severity of asthma in these mice. And using mixed bone marrow cameras, we confirm that this is due to the intrinsic activation of the androgen receptor by testosterone within ILC2s. So testosterone can inhibit testosterone through androgen receptor activation can inhibit ILC2s frequency, but also the capacity to produce cytokine and therefore lung-associated inflammation. So there are different ways to activate ILC2. So here are listed some of the ways. So I showed you before that we activate ILC2s by injecting IL-33 in the mice, and that indeed we can inhibit, we can have the androgen effect when mice are given IL-33, meaning that when male mice are castrated, we have more ILC2s. But we wanted to know whether the androgen inhibition of ILC2s was only seen during an IL-33 type of inflammation, or whether this was also true when we activate IL-33 through other means, for example, through IL-25 or through the IL-2 receptor. And what you could see is that androgen can inhibit ILC2s not only when they are activated with IL-33, but also when they are activated with IL-25 or IL-2 receptor. So what it means that we think that we are dealing with a general way, a global way of inhibition by androgen, and that is not selectively specific to a receptor. So to understand how testosterone was inhibiting how testosterone was inhibiting ILC2s, we decided to move to an unbiased approach. So what we did is that we sorted ILC2 progenitors from the bone marrow of B6 mice, and those ILC2s, we are able to expand them when we add, for example, IL-7 or stem cell factor and IL-33. And this culture has been done in presence of androgen receptor agonists, so dihydrotestosterone, an agonist which is ostarine, which is a non-steroid agonist of the androgen receptor, or in presence of androgen receptor antagonists. So either we activate the androgen receptor, or either we inhibit the androgen receptor. And 7 days after the culture, we analyze ILC2's development. So here, this is the gating strategy to sort the ILC2 progenitors in the bone marrow of those mice. So we knew we could do that because we knew that also in vitro with androgen dihydrotestosterone can inhibit ILC2's development. Indeed, we knew that when we took we did this cell culture for 7 days in presence of dihydrotestosterone, we knew that we can inhibit the ILC2 development by when we add testosterone in the culture. However, when we add fruitamine, so the antagonist, we can push, or at least we can we have the development of ILC2 that is similar to the culture done without androgen receptor agonist. So we know that in vitro we can mimic the inhibition of androgen and that's why we decided to use this system to do our transcriptomic approach. So this is those cytometry data. This is just to show you that in wild-type mice, when we treat the mice, the cells with dihydrotestosterone, we have a type of ILC2s that start to emerge, and those cells, they express, as we could demonstrate in vivo, in vitro, they also express KLRG1 and PD1. So this is just to show you that this model we use, the in vitro model we use to evaluate the ways androgen receptor inhibit ILC2s seems to be okay, as it mimics what we've shown in vivo. So as I mentioned before, so we sorted ILC2 progenitor, we cultivated those progenitor in presence of androgen receptor agonist or antagonist, and we did a single-cell RNA-seq to analyze the transcriptomic signatures of those ILC2s. So those are the clusters we obtained so we did site-seq experiments. So basically we analyzed the cells all together and when you did site-seq experiment, you can differently track the cells. So it was a way for us to analyze better the cells in our single-cell RNA-seq experiment. So those are the cultures. The clusters we obtained. And when we analyze where are the cells treated or not with dihydrotestosterone, we could see that the cells that are not treated with or treated with androgen receptor antagonists, so cells where androgen receptor are not active, they are clustering together. However, now the cells treated with the androgen receptor agonist, so the dihydrotestosterone or the ostarine, they are clustering together. So it means that when androgen receptor is activated, there is a specific transcriptional signature within ILC2s treated with the androgen receptor agonist. And the changes is done in all ILC2s, meaning that all the ILC2s express the androgen receptor and can respond to androgen receptor activation. So those are the genes that were up or down regulated in the different conditions. So so those are the list. And what was striking to us is that when we identified by IPA the pathway that were differently expressed or that were at least induced upon androgen receptor activation, we saw that there was proliferative pathway, but also a lot of repair pathway associated with senescence pathway. So at first it was a bit difficult to understand, because in vivo what we see is that there is that in male, so that when the androgen receptor is active, we have less ILC2s, the cells, they proliferate less, and we have less ILC2s in frequency and in number. But now in vitro, when we have the androgen receptor that is activated, we have we have pathway associated with proliferation. However, when we thought a bit more of the results, we have a pathway associated with proliferation, but we also have pathway associated with reparation and also senescence. And when we analyzed a bit more pathway associated with the exhaustion and senescence, we could see that indeed androgen receptor agonists, they are pushing the ILC2 through a senescence exhaustion signature. So this was highly significant. And when we checked for some of the markers that are supposed to be increased in senescent cells, such as P21 or CD27 that is supposed to be down, and P21 that is supposed to be up, indeed this was fitting. So what we think is happening, we think that that androgen receptor signaling is pushing the IC2 through a senescence exhaustion pathway. And as I mentioned before, we knew that KLRG1, which is a marker associated with exhaustion, is highly expressed in those IC2s. So what we think is that the androgen receptor agonists are pushing those ILC2 through the senescence pathway, and now the ILC2s, they are no longer able to proliferate when they have to deal with an allergen or with an allergen, for example. So this is something we are currently checking in vivo. We are currently checking whether in vivo androgen is also imprinting a senescence pathway in vivo ILC2s, and whether this could have an impact on asthma development, but also other type of inflammation in the lung, such as fibrosis. So this is to summarize my first part. So I show you that testosterone is a powerful inhibitor of ILC2s. Testosterone is able to inhibit ILC2s both at steady state and at inflammation. We've shown that testosterone is also inhibiting the ILC2s development. And we think that through the inhibition of ILC2's proliferation and development, and also their capacity to produce cytokines such as IL-5 and IL-13, this could at least explain sex differences in asthma we can see in a murine model. So, okay, this was beautiful. This is all done in mouse models. So we decided to check whether this was also true in humans, so in asthmatic patients. So indeed, there are in the literature some clues that testosterone is also important for human asthma. Indeed, it has been shown for examples, that in women, when you give women DHEA, which can be metabolized into a testosterone, this has been shown to be beneficial in asthma, in asthma model. It has also been shown both in men and in women that people having increased level of androgen, this is associated with protection in asthma. We also know that there are patients that are deficient in androgen receptor, there are androgen receptor deficiency, and it has been shown in this patient they have an increased risk of developing asthma. They have actually a 4 times higher risk of developing asthma than in the general population. And it has also been shown in a recent paper that there is a SNP in an enzyme here that is able to that is associated with dihydrotestosterone development, and that it has been shown that a SNP which is increasing DHEA metabolism and is associated with a better prognosis of asthma. So there was some hints in the literature that that androgen can be also beneficial in asthma model, but is due to ILC2s and to the impact of ILC2s. So what we decided first to assess is to evaluate whether, as we've shown in mouse model, are there also sex differences in ILC2's frequency in asthmatic patients. So we tested the frequency of ILC2s in male and female asthmatic patients from Toulouse, from Hôpital Larrey. So we obtained the blood of those patients, and we analyzed the frequency of ILC2s in their blood. So this is the gating strategy we use to analyze ILC2s in blood in the patients. And what we could show is that we could confirm what we have shown in mouse, basically, that there is an increase of ILC2s in the blood of women with asthma. This is especially true when in young patients, so between 19 and 35 years old. And these differences was not significant when we analyzed people over 40. So we think that is that is something that is so during this period, we think that there is that estrogen and androgen are present, and we think that this is an hint of maybe a role of hormones. And what we also could see is that these differences was even more pronounced in uncontrolled in patients with uncontrolled asthma, so in patients with severe asthma. So this is showing that there is also sex differences in ILC2 frequency in patients with asthma, as we've shown in mice. But are those ILC2s expressing the androgen receptor? So at the beginning, this was a bit difficult to do. So in ILC2 from the blood, so we decided to move to another type of source of ILC2s, so the nasal polyp. So indeed, the nasal polyp are the frequency of nasal polyp, the frequency of ILC2s of nasal polyp is 10 times more than as compared to the blood. So it was easier for us to purify those ILC2s. So we purified the ILC2s from the nasal polyps, and we try to amplify them with a cocktail of cytokine here, so IL-2, IL-7, IL-33, and IL-25. And you could see that at the end of the culture, we obtained a pure population of ILC2s, so expressing the expected markers of ILC2s in humans, so CD7, CD161, GATA3, and they were not expressing CD3. So we first analyzed whether the androgen receptor gene was expressed within those ILC2s. So we did that by a single cell RT-qPCR. So we first analyzed whether we kind of confirmed that we were dealing with ILC2s, because most of the cells were expressing the GATA3, so the transcription factors for ILC2s. And when we analyzed the androgen receptor gene, we could show that most of those ILC2s were expressing the androgen receptor gene. And so this was all good, but we decided to move forward and to see whether we could, those ILC2 were also expressing the androgen receptor, the protein for androgen receptor. So I'm showing polyp, so nasal polyp from female, nasal polyp from male, or blood ILC2s from male, because at the end we managed to amplify ILC2s from the blood. This is not easy, but we managed to do it. And we could confirm, so if we can look here, for example, a nasal polyp 2, that the ILC2s from human, they express the androgen receptor. Indeed, you have few androgen receptor expressed without the hormone. This is known that in human, the androgen receptor is stabilized with the hormone. So dihydrotestosterone can stabilize the hormone. And also ostarine can stabilize the hormone. So ostarine is a as I mentioned before, is a ligand for the androgen receptor, but is something that is a SARM, or a selective androgen receptor modulator, and it's a molecule that is able to induce androgen-mediated effect, but with less virilizing effect than the dihydrotestosterone. And I mentioned that because I will come back to that later. So we managed to show basically that we can identify circulating ILC2s, and we showed that, as we could demonstrate it in mouse model, that there is an increased frequency of ILC2s in female patients compared to males, that the sex bias was even more pronounced in younger individuals, and also in patients with severe asthma, with severe uncontrolled asthma. We could show that the majority of inflammatory ILC2s from polyp express the androgen receptor genes, and we also confirmed that ILC2s from nasal polyp, but also from the blood, can express the androgen receptor protein. And what we are, so this was through the stabilization by the androgen receptor ligand is something that is suggesting of an effect of the androgen receptor with ILC2. So far, we haven't shown that the androgen receptor is also inhibiting ILC2s in patients. So this is something we are currently doing, trying to understand if dihydrotestosterone, but also ostarine, is inhibiting ILC2 proliferation in vitro, or ILC2's capacity to produce cytokine. And what we are also currently testing is whether ostarine, which is again a SARM that can mediate androgen effect, but without virilizing effect, so which could be maybe more easily used in the clinic, could be does some inhibiting ILC2s in asthma model in mouse, with maybe a therapeutic potential of this SARM in asthma treatment. So I'm done with the presentation. I just would like to thank the people who did this work. So this work has been done for Eve Blancard, who was a PhD student in the team, and now Nina Anesi and Antonin Langlais. So they did a great job dealing with mouse model, but also with a human sample, which is not also always easy. And two great technicians, and Marina Lis, our bioinformatician. And I would like to thank also all our collaborators and the facilities from our lab. So thank you for your attention, and I'm looking forward to your question. Thank you.
Thank you very much, Sophie. It was a really exciting work. I'm really impressed, but a little bit concerned as well, because we haven't paid too much attention to sex differences, right? So
Exactly.
literature is really mostly focused on male experimental models, and we probably missed something.
Exactly. Yeah.
So we are going to first open the floor for questions from the students. I don't know if we have any students that would like to open the microphones and ask questions. Otherwise, we have questions in YouTube, right? Oh, okay. No. So we haven't so far. So I'm going to start asking a few questions, because I'm really curious. The first one is that these findings also apply to other type 2 reactions in other tissues, like parasite infection.
Exactly. Yeah.
For example. Oh, you were expecting this one, I'm sure.
No, because we were working on parasite infection as well, so I didn't have the time to show you, and those results are not published, but exactly. Well, this is something we are currently looking at, because we know that there are sex differences in parasites, and that men, they are more infected, or at least the capacity to eliminate parasites are less effective than in women. We are also evaluating whether this is due to ILC2s, and especially a subtype of ILC2 that are called inflammatory ILC2s, and that have been shown to be key for parasite elimination. Yeah, indeed.
Right. And the other question that I have is that the models were developed mostly in male mice.
Yeah, exactly.
And, the models were adjusted to give us the response that we were looking for. But how much the findings that we actually from these models really can be applied to not only females, but also males in a natural setting that you would expect them to have a lower response. How much the what we have been doing can be compromised by how we have adjusted the model to actually get a response from these male mice.
No, it's true. It's true that this is why basically what we try to do is to test. So I've shown you one type of model we are using, so the HDM model. This is why we are most of the time testing different type of allergens. We are testing different type of background. So we've done B6 mice, but it is also true for BAB-C mice. This is not true for one single type of background. And I mean, this is why we have to move to a human model, I think, now to really prove that this is applying, because this is why we are doing science, to see if this is also this can have any application in human disease. So I think this is why we have to combine all these strategies. Yes, indeed. Yeah.
Right. I don't know if I got it right. You don't have the same sex difference in BAB-C?
No, we do.
No, you do. Okay.
Yeah,
all
this
right.
is why we are testing it. We are testing
Right.
in different type of background. Yes. Yeah.
Okay. Thank you. We do have a question from Median Wernecke here.
Thank you. Hi, Sophie. Thank you very
Hi.
much for your talk.
Welcome.
It's always amazing to realize how little we have been able to explore on things that we thought we knew a lot, right? So I have a couple of questions. One of them is related to aging,
Yeah.
and hormones variations and changes, and the other one is therapy. Basically, we especially women do hormonal therapy as they age, so they age better, right? We age
Yeah.
better, basically. And I wonder if you are able to test things like that, like if in males you give more testosterone, if it reduces even more, because we know also sometimes men use it to get stronger, right? So I wonder,
Exactly.
when we play with hormones,
Yeah.
what happens to these cells?
So with ILC2s and aging, actually, this is something we are doing at the moment to see if because we know that there are sex differences in immunity during aging, and that aging is basically that it's worse in men, and we think this is something due to testosterone. Papers have shown that this might be explaining the reduced capacity of T cells, the CD8 T cells in male to be effective, for example. But regarding therapy in women in postmenopausal women, at some point in the lab, what we were studying the plasmacytoid dendritic cells. And we know that plasmacytoid dendritic cells, that you have to have estrogen for them to be potent. And those plasmacytoid dendritic cells, they are really important to fight infection, to fight and so on. So what we have been able to do is to treat postmenopausal women with dihydrotestosterone. And what we have shown is that by treating postmenopausal women with dihydrotestosterone, you can have now similar capacities of PDC to produce type 1 interferon as young women. So I think this is something important. I mean, something in favor for the treatment with hormone in women, and I think this is something really key, something to push forward, basically. Yeah.
Thank you.
I have actually a few questions. I was very excited about to talk. So the one thing is, the first thing is a clarification, because you actually use testosterone in the in vivo experiments, but the in vitro you use dihydrotestosterone.
Yeah.
Is there a role for 5-alpha reductase in vivo?
Is there, sorry, can you, is there what in vivo?
I was wondering if you actually have to have the conversion from testosterone
that's why, exactly,
to dihydrotestosterone.
Yeah,
and,
that's why.
That would happen in vivo, but not in vitro, and that's why you
Exactly, that's exactly why. Exactly. If you put testosterone in vitro, you have no effect at all. I mean, you have to have the conversion before. So and that's why we put the hydrotestosterone in vitro, basically. Yeah, exactly.
Well, that's really interesting. You know, we are talking about the same receptor, but it has such amazing different sets of effects depending on the ligand. I find it really fascinating. We have a question that was posted in the chat in YouTube that is from Camila Batista, saying that it's a simple question, but when you look at the animal model, do you observe differences with the female estrous cycle?
So this is really difficult to do, because basically in female, the cycle is really short, it's 3 to 4 days.
Right.
And I don't think estrogen has an effect on females' ILC2s, because we've done ovarectomy, we've tested models in which you have the estrogen receptor deleted in the hematopoietic compartment. I don't think it has an effect, really, but it's tough to do, because for other experiments, for example with PDC, we have done that, studying the frequency during the estrous cycle. This is tricky, but it can be done. I think for ILC2s, it might not be a problem, but for other type of cells, it has been shown that indeed you can have a variation during estrous cycle of the function, but also the frequency of the immune cells. So yeah.
Right. Thank you.
It's complicated.
Going back to the first part of your presentation with the X-linked genes, you talked about toll-like receptor 7. I was wondering if there are other genes, and especially ones involved with type 2 immunity that may be regulated this form. And I ask that because I know that GATA1 is an X-linked gene, and it's really important for eosinophil development. So I was wondering if you have any data that may really play together with the hormones regulation and the XX individuals.
Yeah. So for the ILC2s, it's a good point. We haven't looked at GATA1. What we have looked at is with a special focus on trying to understand SLE, sex bias. We have demonstrated that TLR7, but also TLR8 can escape X chromosome inactivation, so in B cells and in monocytes. And other groups have shown that there are some, for example, there are some genes linked to TH1 function that can escape, but regarding ILC2s, we haven't, no, we don't know whether there is like a combination of both factors that could explain this effect. No, we haven't done that so far. Yep.
Right. Thank you. We actually have 2 questions in the Zoom room. The first one is from Barbara. Great presentation. I do have a curiosity. Have you considered looking into transgender men and women and how the hormone treatment could affect asthma and ILC2 populations?
Yeah. So actually, there are some cohorts that are starting to be. I mean, we can start to look at those the cohort we are now. So that there have been a paper published by Peter Brodin recently in Nature or Science, I don't remember, and they have a cohort where they analyze PDCs because plasma cytoid dendritic cells, because there is a huge bias in PDCs, and we know that estrogen can really have an impact on this population. So they demonstrate that indeed in transgender cohorts, so you have differences. You can mimic the differences, the sex bias by giving the hormone basically. So for ILC2, there is nothing known. We don't know yet. We are starting to think about doing it, especially because in Toulouse we have a cohort of transgender that have been given testosterone. So that could be a good point to look at ILC2s, and we are starting to explore that actually. Yeah.
Right, and we have another question from Ian Antunes. He says, thanks for the presentation. I was thinking about the possible side effects. Have any of these been observed in experimental models? This came to mind when reading about the use of the specific androgen receptor modulators, in asthma, especially in the context of childhood asthma, as I have had asthma since I was a child.
Yeah, so of course this is really, I mean, this has to be taken really with really great question. I mean, what has been done so far is treated women with DHEA, so like and this has been done in 2 clinical trials so far. But for example, giving Ostarine to women has never been done so far. So this is something we would like to test. Ostarine is actually a treatment for muscle diminution with cachexia. But I mean, so this is something first we are going to test in. This is not the first treatment of choice, of course, but
of course. But have you ever thought about having it in inhaled form so it could limit its effects to the lungs?
Yeah, of course. The DHEA, it was given. It was only aerosol. It was So it will be also the case with. But maybe you can have some that is going through the circulation and But Ostarine, it is given in quite a lot of patients. You have really less virilizing effects, so you have less air growth and so on. So that could be an option. Yeah.
All right. I think we have exhausted our questions, not our curiosity about it, but only our questions. Thank you very much.
You're welcome. Thank you very much for the
wonderful
invitation.
talk. Thank you for accepting our invitation to be here today. And for everybody else, we hope that we see you next week, same time, same channel. Thank you very much, Dr. Sophie Laffont.
Bye-bye.
Au revoir. Merci beaucoup.
Merci.